Much for joining the. Thank you so much, for this wonderful gathering. I take immense pleasure and honor in introducing Dr. Milind Ratnaparkhe, who is a senior principal scientist at IISR, Indian Institute of Soybean Research in Indore, Madhya Pradesh, India. I've had a very good fortune in knowing Sir over the last, I would say, 2 decades. And that was my first recall. I still remember in 2004 when I was just a PhD fellow. We were a part of the growing Genome India International community pronounced by Professor Chittaranjan Kole. And I would probably ask very iota of questions because Sir was already serving as a senior scientist in University of Missouri in the US then. And he would fervently ask and discuss with me and all those kind of things. Although I was coming from a human biology background and Sir largely from chickpea and plant genomics, he's always kind enough, always smiling, and always a kind of a great ambassador for Genome India International over the last 2 decades. Just to have a quick introduction, Dr. Ratnaparkhe obtained his MSc from Devi Ahilya University in Indore from 1991, and then he went on to do his PhD and obtained in 1999. After which he moved to US for his subsequent postdocs and work for University of Missouri and several other universities. And he has had a large interest focusing on recombinant inbred lines in chickpea, for which he has used fusarium wells, for which his largely cited paper was seen some 12, 13 years ago with more than 300 citations. And then with a large interest for India and for our motherland, he returned back in 2012, where he has taken up as a principal scientist at Soybean Research Center. And Sir has been, as I told you, has been an ambassador for Genome India International, serving as vice president and secretary in several other portfolios. Thank you so much, Milind sir, for your gracious time and presence today. The floor is yours. Yeah.
Thank you very much, Prash, and good afternoon, everyone. It's really a great pleasure to be here with the group of Genome India International, and I really thank all the organizers and the committee to giving me this opportunity. Today I would like to share about my views on the crop, soybean crop. And as we all know, it's a very important crop globally, and from like several countries like US, China, Brazil, Argentina, and India are a leading soybean producing countries, and it has a wide application. So a lot of research already done in soybean at international level globally. So I'll briefly discuss about the work what we are doing here. Among the oilseed, if you see, there are a couple of different oilseed crops, but soybean is among the top oilseed crops globally. And other crops are rapeseed, mustard, groundnut, palm oil. But more than 60% of the edible oil in our country is imported, and we are not self-sufficient in producing the sufficient oilseed crop. So there is a huge demand for edible oil, and a lot of foreign exchange goes in procuring this edible oil. And currently there is in India a lot of focus to increase the oilseed production. And if you look at the soybean, it has multiple use for food, feed, as a biodiesel, fire nutraceuticals. And recently I attended a conference for animal feed. And there is a huge demand for poultry industry, for fisheries, for cattle feed. And this soybean is widely used and there is shortage. And what is happening is in India, the crop oil is extracted, and the remaining, after extraction, the remaining part is exported as a feed to several countries, European countries. And our farmers, they get a special earning after exporting that feed, animal feed. Regarding the human consumption, very less human consumption in India, but in other countries, soybean is widely used as a food. If we look at the area, production, and the yield, in India, it is grown in mostly three states: in Madhya Pradesh, Maharashtra, and Rajasthan. These are the three states which cover more than 80% of the soybean. And the total area is around in India is 12 million hectares. And the production, yearly production differs from 13 to 14 million metric tons. So this is a very significant area which we have on soybean production. And the soybean, if you look at the as a crop, this was not grown in India before. It was mostly grown in the northeast region and in the Uttarakhand region. They were using for food consumption. And later in 1970s, the area spread in Madhya Pradesh, Maharashtra, and Rajasthan. And in a very short period, around 12 million hectares were grown. So this is a very fast this area was developed. And if we look at like in 3 decades, this area of 12 million hectares was produced. If you look at the 5 years production for soybean, so it is almost stagnant. And again, the yield is ranges from 10 to 12 million tons. And there is a huge demand to increase the production and productivity. And how we can develop new technologies so that we can increase the production and productivity of soybean. So that is a very important part. And here we are trying our best to increase the production and productivity by developing new varieties. So state-wise, I have already mentioned that Madhya Pradesh, Maharashtra, and Rajasthan are the major state, but there is a huge possibilities in the southern India, in Karnataka, in Andhra Pradesh. A lot of farmers are interested, and also in Uttar Pradesh also. So a new region where the soybean can be grown is expanding, and we're hopeful that new varieties can be developed where in India in other states also soybean will be more popular. Now there are a lot of major challenges as we all are aware about the climate change and abiotic stresses, especially drought, is a very big challenge. In central India, what we have seen that after the sowing of soybean, there is a dry spell for 15 days, 20 days, and immediately after sowing, there can be a drought stress on the plants. Sometimes even during flowering or seed filling stage, there is a long dry spell and drought stress is there. So drought is a very big issue in soybean production. And mostly the soybean is grown in rain-fed conditions, like irrigation is not given. So whatever we get the rainfall, that water is used by the plants, and therefore there is no special irrigation given to this crop. There are also certain states which have more water rainfall, and waterlogging is also a problem in those states. Chhattisgarh, in Odisha, if there is a more rainfall, so that is also an issue. There are a lot of biotic stresses. Like last couple of years, we have seen that some new fungal disease, new viral disease are emerging in the areas which were not there before. So biotic stress is also a very big concern, and we have to work on identification of the diseases, insect pests. We have to identify those genes which provide resistance to diseases, and therefore genetic mapping and genomics for both biotic and abiotic stresses is our top priority. And in addition, there are other challenges like it's a rainy season crop, so a lot of weeds are there. So herbicides are also used for controlling the weeds. So with this major challenges at our institute, different groups are working to improve the new soybean varieties. Now, if you look at the genomics part, the first focus is on germplasm characterization. And globally, huge germplasm is available at different countries. In India, around 4,000 lines are available at our institute. And these germplasm lines are extensively characterized for various traits, for disease, for drought, for water, like for flowering, then seed composition trait, how much is the oil percent, protein percent, and to improve the oil quality. So we are using these germplasm, but recently from US we have again imported huge germplasm, and which is under quarantine currently, and that is also available for our studies. So germplasm characterization for various traits is very important. Second is definitely identification of the molecular markers. So we identify the genes and markers. So these markers are used in the marker assist breeding program to develop the new varieties. So this is a major aim, and with the development of genomics, translation genomics is being used. So soybean genome was sequenced in 2010, and currently a lot of genomic resources are available, like reference genome. At least 3 reference genomes are available, and around 2000 whole genome sequences of soybean lines is also available. So a lot of genomic resources available for improving the soybean. For germplasm screening, as I mentioned, we have 4,000 soybean lines, and this we screen for different abiotic stresses for drought tolerance, waterlogging tolerance, and also we screen during summertime. And then we developed a panel which is highly diverse. So this 300 lines was finally developed, and these lines we have used extensively for phenotyping and genotyping. So here I would like to mention that there is a collaborative project with the ICRISAT and also at National Institute of Abiotic Stress Management, Baramati. And with ICRISAT, we collaborated with Dr. Rajiv Varshine and Ms. Anu Chitikeni. And at Baramati, Dr. Ajay Singh is the collaborator. So last 4 years, we have been working on these 300 soybean genotypes for high throughput phenotyping, sequencing, and we are looking forward for identification of new genes, new markers for different biotic and abiotic stresses. So this is just a picture showing the screening of the plants for drought tolerance. So we have these rainout shelters where we control the water requirement water, and on the left side is a control. So this is completely having irrigation and full water. But on the right side, we give a stress at a particular time. That is when there is a flowering or seed filling. Around stress is there for 20 days or more. And after that, we measure a lot of physiological parameters and yield related traits. And these are then used for the molecular studies. So this is the phenotypic data. On the left side, if you see the large number of drought-related traits are observed, like seed yield, then 100 seed weight, plant height, number of nodes, then chlorophyll content, relative water content. So like this, we take a lot of traits after giving the drought stress. And on the right side, key genotypes are identified, which are very good for control are having drought stress, and these lines are later used in the breeding program for developing the new varieties. So extensive screening for this drought stress has been done. And if we look at the like different traits, this is a control and another is stress plant. So there is a reduction in the biomass, also harvest in index, also in the stress condition, it is reduced. Number of nodes also we can see the differences, number of pods also. And this data is very useful to pinpoint the genes associated with these traits. So we are in the process of identifying genes for large number of traits for drought. This is another area is the water logging, and this is a field where we are growing these 300 lines. So what we do is that when the plants are in the vegetative stage, then we give this stress around 12 to 15 days water logging stress is given. And again, we measure a lot of different parameters and physiological traits associated with the water logging tolerance. And we have recently identified very good lines which are having good tolerance for waterlogging, and the breeders in our institutes are using these lines for development of the new varieties. These are some of the traits like foliar damage score is there, then what is the plant mortality rate, then again seed weight, percentage of the seed weight loss after the water stress, and percentage loss in plant height. So a lot of traits identified. And these genotypes, again, as I mentioned, are being used for developing the new varieties. Here also we can see the differences after giving the stress, differences in the stress plant and the control plant. And these are being used for association, genome-wide association studies to identify the genes. Another area is the root system architecture, and this is also a very important trait. So what we do is these are the pipes which are 6 to 7 feet height pipe, and soybean is grown here. And after some time, we cut this pipe, open it, and the roots are removed very carefully. And then we do scanning for these roots, and then we take a lot of measurement for root length. So this was experiment was done for last 3 years and very laborious work. And we have recently completed this root trait architecture phenotyping, and now this is ready for further analysis for identification of the genes. So this is a variation what we see in the root trait architecture. Some of the plants have very long roots, some have root angle is different, some root density is also different. So a lot of variation in all these soybean lines. And this variation is very important if you want to look target for any gene or for any SNP which is associated with the trait. So these are the again the root system architecture, root depth, then root length. All these parameters are measured. Root volume. Then there is a scanner which we have in our institute, and these roots are then scanned on, and then all the data is recorded. So this data is will be again very helpful for us to pinpoint the genes associated with these traits. So we have identified large number of genotypes with a very good root traits, and these we are hopeful that these lines will be used by the breeders in crossing program and for developing the new soybean varieties. Now this is the overall picture what genomics assisted varietal development soybean we are going through. So we have a GWAS panel, and we are doing these association mapping studies, and currently working on haplotype identification and gene identification, also pinpointing the SNPs. So for all these traits, in addition to drought, we are also working on early maturity. This is a very important trait. What is happening is in India, farmer prefers the soybean varieties which grows only 90 days. If the variety is more than 90 days, 100 days, 120 days, then that variety is not adopted, because they want to go for another crop immediately after the soybean is harvested. They want to grow either potato or wheat or chickpea, or different crops are. So maturity is very important, and the early maturing soybean varieties are required. So large number of genes associated with the flowering have been identified, and this is also a photoperiod sensitive crop. Like the variety which is grown in the central India will not will not produce much yield in other states in southern India or in the northern India. So these a lot of genes are there related to photoperiod, and we have to see that these varieties which we are developing here are also grown in other places. Similarly, heat tolerance is also another important trait, and we are also working on this. So in addition, for functional genomics, we are working on virus-induced gene silencing, and this work, a lot of good progress has been made in weeks. Some genes have been taken, and these genes we have silenced, and then found that these genes play a very important role for drought tolerance. So our future aim is to develop the RNAi lines or genome edited lines. After these genes are identified, we are going to do functional validation from WIX and RNAi. And in the near future, again, we are initiating genome editing for large number of traits for abiotic tolerance, biotic stresses, and seed composition traits. So as I mentioned that the 300 lines of data is already available, and that we are using for several different traits. So I'll briefly discuss about This is the diversity studies and of these 300 lines, and the SNP data was used for estimating the diversity, and we have a very good collection based on the molecular data. Also, now we can say that these lines are highly diverse, and they can be used for different traits. So we are also taking the same lines as the data is available. We are working on charcoal rot resistant, anthracnose resistant, YMV. These are the other diseases where the same panel is being used for phenotyping, and this will be used to pinpoint the genes for different diseases. Now, as I mentioned, like a lot of whole genome sequencing data is available. And what is our plan is that this data of 300 lines, we will be comparing with around 20,000 USDA lines, because that USDA data, SNP data is available. So we can find this relationship between the local germ plots available here and at the globally, at the international level, what is the genetic relationship, and the whole genome sequence data also we are comparing, and that will be used for our diversity studies for pinpointing the genes. Now, in functional genomics, as I mentioned, that virus-induced gene silencing and RNAi are the two approaches, and Dr. Singh at Baramati has taken a lead, and the vector was patented in US, and the same vector we are using here for this virus-induced gene silencing studies. So this is one experiment which was done for overexpression of the FAT3 gene. And what we found is that it enhances drought and salinity stress tolerance in soybean. So here you can see the on the top, the drought stress, which is overexpressed FAT3 gene. This FAT3 gene is associated with the oleic acid content. And when we overexpress this gene, then there is a increase in the drought stress tolerance. Similarly, this was also found for salinity stress. This is another area where we are working. So the gene was has played a very important role in giving abiotic stress tolerance in soybean. So we are still working and confirming these for other abiotic stresses. And this work was published in Plant Molecular Biology recently. And we are hopeful that we will be able to develop the drought tolerant lines using this FAT3 gene. Another is silencing of farnesyl transferase gene. And here also we found that silencing has induced the water stress tolerance in soybean. So this is the first time again a new gene is being reported. And these genes were initially identified from Arabidopsis, where these genes were confirmed that they play a role in abiotic stresses. And from orthologs from Arabidopsis were used and in the soybean gene was identified, and this gene was silenced. So again, after this virus-induced gene silencing, we'll be moving ahead with the RNA approach to develop the transgenic soybean lines. This is another gene, into gene, and silencing has also improved the this drought stress tolerance. And so all these three genes are candidate genes for abiotic stress tolerance. But in addition, we are also planning for several other genes based on our genome-wide association studies. New genes are being identified, and those genes we will do silencing and then confirm their function. And in future, we will do plan to do genome editing to improve the abiotic stress in using these genes. So the genomic resources, as we discussed in other countries, more than 1,000 whole genome sequences are available. Currently, also even up to 2,000 whole genome sequences data is available. SNP data of 20,000 soybean lines is available at the USDA, and large number of marker associated with the important traits are available. But again, still there is a big opportunity to use this huge data for developing new markers and for varietal development. And we are using those high throughput genotyping approaches to in the marker assisted breeding program. I would also like to discuss about the different diseases, and this is soybean rust is a very important. Another are charcoal rot, anthracnose, YMV. I was working on this soybean rust for a last couple of years, and what we found is that in the Maharashtra there is a region where this rust occurs every year. So that is a hot spot for soybean rust, and worldwide this is a very important disease. So it is important to identify the genes. Currently, there are seven genes are known for soybean rust, and these genes are race specific or isolate specific. So what we started working on, there was one line which was found to be resistant. Like we screened several germplasm in the southern India, in Dharwad and Sangli region in Maharashtra. And what we found is that one genotype EC241780 is having resistant. And on the other side, you can see this is a susceptible cultivar. This JS335 is a very widely grown cultivar. Last 2 decades, this has been a very prominent variety. And in Maharashtra, in southern India, this is widely used. But this is highly susceptible for this disease. And the farmers have to spray fungicide to control this. But the effective way is to transfer these resistant genes. So what we did, that whole genome sequencing of this line was conducted, and then we pinpointed the RPP1 gene, which is present on chromosome 18. So that gene we pinpointed, and now that markers have been developed, and the breeders can use this marker for developing rust resistant line. This is the SNP data which was used for identification of the marker and gene. So on the left side, all are the rust susceptible lines, and the arrow indicate the lines which are tolerant. So the soybean line which we sequence in India and the other line in US have 100% sequence matching in that region where the rust resistant gene is present, and this is present on chromosome 18. So by just comparative genomic studies, we were able to identify the location of the gene. And now since this gene is known, we are developing marker based on the genic information. So this is the information regarding these three genes are there, which are very similar to each other. When we do multiple sequence alignment, they show very high similarity. And these are called as the NBS LRR genes. Nucleotide binding site, leucine rich repeat genes are there. So this is the first line in globally. This is the first line which was sequenced for rust resistance, and huge data is available with us at different regions. And at the bottom, you can see there are some insertions and deletions. So within these three genes, we can see insertion of 3 nucleotide, which is adding one amino acid the protein sequence. So we want to see how by addition of this one the amino acid, what is the difference in this protein, and how it is governing the resistance. So these minor changes are there in the tolerant and the resistant lines. So these insertion deletions are being used for developing molecular markers. And so these markers are present within the gene, and so they are very effective in transferring the gene to the susceptible line. And this was one paper which we had published about whole genome sequence of soybean accession, and it involved in the rust resistance. Another area is this charcoal rot resistance, and this is another disease. So what we have found that if charcoal rot comes, the entire the field is gets infected, and the yield is there are huge loss for the soybean yield. So phenotyping was developed, and here we can screen the large germplasm through our phenotyping based test, and this we are using for genetic mapping and genome-wide association studies. So by GII studies, we have again pinpointed certain chromosomal regions. If you see this chromosome 8, 9, 11, 17 are showing the SNP markers which are promising, and this area we are still are exploring to identify which is the gene present on these chromosomes which can play an important role for charcoal rot resistance. So like this, we are doing GII studies for different diseases and pinpointing the genes. These are some of the putative genes which has been identified in soybean for charcoal rot resistant at seedling stage. So this screening was done at a very early stage, seedling stage, and a large number of genes are there. But again, to validate these genes, we have to go for functional genomics or gene expression studies. And using RT-PCR, gene expression studies are done to confirm that really this gene is associated with the charcoal rot resistance. So, so for these after the gene expression studies, we develop confirm and develop the markers. So with this, I just would like to stress that our work on biotic stresses, abiotic stresses is going on to identify different genes and markers for drought tolerance, waterlogging tolerance, unique root trade architecture. For seed composition, oil content and protein content also we have done data is available, and we are also working on it. But as I mentioned that there is a big challenge to develop the varieties which are high yielding, which are having early maturity, are resistant for different diseases, and also for abiotic stresses, drought, waterlogging, etc. And therefore, but based on the available resources at the genomic level and the new advances in genotyping facilities, we are very hopeful that in soon we will be able to develop new varieties with these traits. So with this, I would like to thank you, and I'll be happy to take any questions. Thank you very much once again for all your joining in this.
Thank you, sir. Thank you for such a wonderful talk. Now the session is open for discussion. If anyone has any question, please raise your hand.
Prash, hi, this is Dr. Rakesh Srivastava from IISR. Can I?
Hello, hello, Dr. Rakesh.
Hello, Dr. Milind. Nice to see you.
Same here, thank you.
Really wonderful presentation. Although I joined late, but I enjoyed your, last few slides. So congratulations, you seem to have done wonderful job, wonderful work. And you might know IISR is adopting additional crops. So soybean is one of them, and we will have opportunities to, collaborate with you.
That will be really wonderful because we are looking forward. IISR has a lot of expertise in legume crops and oilseed crops. So we really want to have collaboration with IISR and other institutes. And currently, as I mentioned, that soybean genomics has been developed at a very good scale. But when we collaborate with different institutes, we can learn and move faster. So I'm looking forward to it.
Yeah, so we want to collaborate with you because we want to learn from you, Dr. Milind.
Sure.
Yeah,
I'm always available.
Thank you. So my question is, if I heard it correctly, you said you want to carry out GWAS, and from your GWAS results, you would like to silence, some of the genes. So my understanding is, GWAS results are kind of positive results. So they give you, peaks for the traits which actually contribute. So when you go ahead and silence them, don't you think you might not move in right direction?
Actually, the silencing, what we are thinking is just to confirm the gene, whether after silencing it is susceptible for drought tolerance like that. It's a very small 30 days experiment using virus-induced gene silencing. So either by gene expression also we want to confirm that that is really associated with the drought tolerance or disease, and by silencing that will give additional benefit. But what is your suggestion in this? Like, what is the best way? I would like to hear.
So yeah. So this is a good concept. So see, for example, if you want to target genes for say charcoal rot, you do GWAS, and you then find out some peaks actually for charcoal rot. Now, when you go ahead and silence these genes, it's quite possible that actually you are doing this for susceptibility to charcoal rot. But then, that will also give you a proof of concept that, that is the candidate gene. And then if you develop a kind of tilling population and, do some tilling by sequencing and are able to, validate this in diverse, genetic backgrounds, I think that's the way to go. I mean, it becomes something like your reverse genetic tool. So,
Sure.
idea is really great. Yeah. Yeah. I congratulate you for that.
Sure. I agree with you. And yeah, definitely we'll be looking forward to this approach.
Yeah. So yeah, incidentally, soybean has been given to me. I work on pulmonary genomics and molecular breeding. Yeah. And the crop which you see in background is a mega cultivar called HHB67 improved 2, grown in more than 10% of the total cropped area. We stacked 3 QTLs, and this was dedicated to nation by Honorable Prime Minister some time back. So I really, yeah. So we would like to replicate these kind of success stories in soybean, and fortunately, I've been given soybean. So pretty soon I'll get in touch with you, Dr. Milind, and it is such a pleasure, speaking to you and hearing you. My congratulations and best wishes.
Thanks a lot, and I'm looking forward to talk to you and move forward in this work.
Yeah. Thank you so much. All the best. Thank you, Prash.
Thank you, sir. Yes, Dr. Abhimanyu Sarkar.
Thank you very much for a great talk, and thank you very much, Dr. Prash, for inviting me, and thank you, Dr. Milind. It was really a very interesting talk. So here at NIAB, that's the National Institute for Agriculture Botany, we are looking to develop soybean for UK. It's not a crop that's traditionally grown in UK, and we are doing some basic studies as well as varietal development. And it's very interesting to hear both from you and also what Dr. Srivastav said, the progress that India has made in soybean development. One of the things that we are, of course, looking at is flowering time traits to adjust the crop phenology to have soybean mature in the growing season. So we are really looking at very early varieties of soybean. Even 000 maturity group soybeans don't really do well in UK. We are looking at developing 000 cultivars. So that's a goal of our project. We would be happy to collaborate with you on that, especially on photoperiodism. But also the other thing is WIGS. I have a PhD student who is looking at developing new WIGS vectors again. And we also work on recombination. So just increasing the recombination frequency using certain genetic systems. So yeah, we would be happy to collaborate with both you and ICRISAT. And yeah, it's really amazing how much progress has been made in the genomics-driven plant breeding in India. After I always look up to Dr. Rajiv Varshney because he sort of I would say revolution in Indian plant breeding. So yeah, it's great to be associated with all of you. Thank you very much.
Thank you very much, Dr. Sarkar. And I'm very glad to know that you are working on soybean. And we are also looking forward to exchange ideas and collaborate in this field. And as you mentioned, like in our institute, photo period is a very important trait. And developing early maturing variety is key for success of soybean in India too. So I would be very happy to personally talk to you and exchange ideas and collaborate. So
thank you very much. Thank you. Looking forward to it.
Thank you, sir. Sir, one question is there in chat box from Dr. Ganpati Mukri. Is FAD gene has pleiotropic effect, as it also known to affect oleic acid pathway?
Yeah, that is this is for oleic acid pathway, and therefore we want to be sure that by silencing or by overexpression we don't change the seed composition trait. So that is a point of concern, and therefore we will move very carefully working on that FAD3 gene. So, but because it is associated with the oleic acid content.
Okay, thank you, sir. Any question? It seems your presentation is self-explanatory, so everyone understands well. So thank you so much, sir, for accepting our invitation, for delivering this wonderful talk. Thank you, sir.
It was a great pleasure, and I'll be very happy to collaborate or to discuss with anyone on soybean. And we are looking forward to with your collaboration in this. Thank you very much to everyone.
Okay, thank you all the participants for joining. Thank you, one and all.
Bye-bye.